Reproduction! Always a popular topic, and one that I don't mind saying that I am personally interested in. The kind of reproduction that we're most familiar with is, of course, sexual reproduction, where sperm meets egg, they share genetic information, and then that fertilized egg splits in half, and then those halves split in half, and so on and so on and so on, to make a living thing with trillions of cells that all do specialized things. And if you're not suitably impressed by the fact that we all come from one single cell and then we become this, then I don't know how to impress you. But riddle me this, my friends. If sexual reproduction begins with sex cells, the sperm and the egg, where did the sperm and egg come from? Aw, dude. So how do sex cells form so that they each have only half of the genetic information that the resulting offspring will end up with? And for that matter, why aren't all of our sex cells the same? Like, why are my brother John and I different? Sure, we both wear glasses and we both kind of look like a tall Doctor Who, but you know, we have different color hair and different noses, and I'm way better at Assassin's Creed than he is, so why aren't we identical? As far as we know, we both came from the same two people with the same two sets of DNA. The answer to these questions, and a lot of other of life's mysteries, is meiosis. Meiosis replicates a cell with a complete set of 46 chromosomes into two daughter cells that are each identical to each other. But of course, even though the vast majority of your cells can clone themselves, you cannot clone yourself. And for good reason. Actually, reasons. If meiosis were the only kind of cell division we were capable of, that would mean A, you would be a clone of one of your parents, which would be awkward to say the least, or possibly B, half of your cells would be clones from your mom and half would be clones from your dad, and you would look really weird. But that's not how we roll. We do things a better way, where all of your body cells contain the same mix of DNA, 46 chromosomes grouped into 23 pairs, one in each pair from your mom and one from your dad. Those pairs of chromosomes are pretty similar, but they're not identical. They contain versions of the same genes or alleles in the same spot for any given trait. Since they're so similar, we call the pairs homologous chromosome pairs. Homologous is a word that comes up a lot in genetics. It just means that two things have the same homo relation logos, even if they are a little bit different. However, there are some very special cells that you have that have only one half of that amount, 23 chromosomes. Those are sperm and egg cells. These are the haploid cells. They have half of a full set of chromosomes. And they need each other to combine to make the complete 46. Creating those kinds of cells requires a process that's very similar to mitosis, but with a totally different outcome. Meiosis. That's when a specialized diploid cell splits in half twice, producing 4 separate cells, each of which is genetically distinct from the others. Meiosis is a lot like mitosis, except twice. It goes through the same stages as mitosis: prophase, metaphase, anaphase, and telophase. But then it goes through another round of those stages again, and they have the same names, conveniently, except with a 2 after them. They're like sequels. And just as with the Final Destination movies, the sequels have pretty much the same plot, just some new actors. And the raw materials for this process are in your ovaries or your testes, depending on, you know what it depends on. Now, just like with mitosis, there's a spell between rounds of cell division where the cell is gearing up for the next big split, and this is called interphase, when all the key players are replicating themselves. The long strings of DNA in the nucleus begin to duplicate, leaving 2 copies of every strand. And that brings us to the first round of meiosis, prophase 1. This is nearly the same as in mitosis. The centrosomes start heading to their corners of the cell, unspooling the microtubules, and the DNA clumps up with some proteins into chromosomes. Each single chromosome is linked to its duplicate copy to make an X-shaped double chromosome. Now keep this in mind, once attached, each single chromosome is called a chromatid, one on each side of the X. Each double chromosome has 2 chromatids. Here in meiosis, prophase 1 includes 2 additional and very important steps: crossover and homologous recombination. Remember that the point here is to end up with 4 sex cells that each have just one single chromosome from each of the homologous pairs. But unlike in mitosis, where all the copies end up the same, here every copy is going to be different from the rest. Each double chromosome lines up next to its homologue, so there's your mother's version lined up right next to your father's version of the same chromosome. Now if you look, you'll see that these 2 double chromosomes, each with 2 chromatids, add up to 4 chromatids. Now watch. One chromatid from each X gets tangled up with the other X. That's crossover. And while they're all tangled up, They trade sections of DNA. That's the recombination. The sections that they're trading are from the same location on each chromosome. So one is giving up its genetic code for, like, hair color or body odor, and in return, it's getting the other chromosome's genes for that trait. And this is important, what just happened here. Creating new gene combinations on a single chromosome. It's the whole point of reproducing this. What's more, since all of the 4 chromatids have swapped some DNA segment at random, that means that all 4 chromatids are now different. Later on in the process, each chromatid will end up in a separate sex cell, and that's why all eggs produced by the same woman have a slightly different genetic code. Same for sperm and men. And that's why my brother John and I look different even though we're both made from the same 2 sets of DNA. Because of the luck of the genetic draw that happens in recombination, I got this mane of luscious hair, and John was stuck with his trash brown puff. And don't forget about my mad Assassin's Creed skills. But what comes next is another kind of amazing feat of alignment. This is metaphase 1, and in mitosis you might recall that all of the chromosomes lined up in a single row, powered by motor proteins, and were then pulled in half, but not here. In meiosis, each chromosome lines up next to its homologous pair partner that it's already swapped a few genes with. Now the homologous pairs get pulled apart and migrate to either end of the cell, and that's anaphase 1. The final phase of the first round, telophase 1, rolls out in pretty much the same way as mitosis. The nuclear membrane reforms and nucleoli form within them. The chromosomes fray out back into chromatin. A crease forms between the 2 new cells called cleavage, and then the 2 new nuclei move apart from each other. The cells separate in a process called cytokinesis, literally, again, cell movement. And that is the end of round 1. We now have 2 haploid cells, each with 23 double chromosomes that are new, unique combinations of the original chromosome pairs. In these new cells, the chromosomes are still duplicated and still connected at the centromeres. They still look like X's. But remember, the aim is to end up with 4 cells, so it's time for those sequels. Here, the process is exactly the same as mitosis, except that the aim here isn't to duplicate the double chromosomes, but instead to pull them apart into separate single-strand chromosomes. Because of this, there's no DNA replication involved in prophase 2. Instead, the DNA just clumps up again into chromosomes, and the infrastructure for moving them, the microtubules, are put back in place. In metaphase 2, the chromosomes are moved into alignment into the middle of the cell, and in anaphase 2, the chromatids are pulled apart into separate single chromosomes. The chromosomes uncoil into chromatin, the crease-forming cleavage and the final separation of cytokinesis then mark the end of telophase 2. From one original cell with 46 chromosomes, we now have 4 new cells with 23 single chromosomes each. If these are sperm, all 4 of the resulting cells are the same size, but they each have slightly different genetic information, and half will be for making girls, and half will be for making boys. And that's all there is to it. I know you probably were really excited when I started talking about reproduction, but then I rambled on for a long time about haploid and diploid cells. But now you can say that you know more about the miracle of reproduction. It's not actually a miracle. It's science.